Suggest and explain how the structural difference of cDNA and genomic DNA leads to only cDNA being expressed successfully. cDNA is made from mature mRNA, so it contains only exons, which are the coding sequences of the gene. In contrast, the genomic DNA taken directly from human cells containing both exons and introns because the gene has not yet undergone RNA processing. Bacteria are unable to remove introns because they lack the splicing machinery found in eukaryotic cells. As a result, if genomic DNA is inserted into a bacterial plasmid, the bacteria cannot produce a correctly processed mRNA transcript, so a functional polypeptide cannot be translated. However, because cDNA contains no introns, bacteria can transcribe it directly into a complete functional mRNA that can be translated into the insulin protein. Therefore, only cDNA leads to successful gene expression in a bacterial host. Section B, Part 1. State the role of DNA ligase in the formation of recombinant plasmids. DNA ligase joins the human gene fragment to the plasmid by forming phosphodiester bonds between adjacent nucleotides in the sugar phosphate backbone. After the gene and plasmid pair at their complementary sticky ends by hydrogen bonding, DNA ligase permanently seals the strands, ensuring a stable recombinant plasmid is formed. Section B, Part 2. Explain why a promoter as well as the gene may have to be transferred into the plasmid. A promoter is essential so that RNA polymerase can recognize the bind to the DNA to begin transcription. Without a suitable promoter sequence upstream of the gene, the bacterial RNA polymerase would not initiate transcription, meaning the gene would not be expressed. Including a promoter ensures that the inserted gene is transcribed and therefore the recombinant protein can be produced. Section C. Explain how a marker gene coding for a fluorescent product could be used. A marker gene encoding a fluorescent protein is inserted into the plasmid alongside the gene of interest. It is placed downstream of a promoter so that it can be transcribed and translated in transformed cells. When the plasmid is taken up by a bacterium or yeast cell, the marker gene is expressed, and the cell produces a fluorescent protein. Under UV or blue light, these proteins fluoresce, causing transformed cells to glow. This allows researchers to easily identify which bacterial colonies have successfully taken up the recombinant plasmid because only these cells emit fluorescence. Section D. Suggest advantages of using yeast compared to using bacteria for human insulin production. Yeast cells offer several advantages over bacteria. They are easier and often cheaper to culture on a large scale, and they typically show higher productivity, allowing more insulin to be produced per unit volume of culture. Yeast cells are eukaryotic and therefore more similar to human beta cells. They possess organelles such as the Golgi apparatus, allowing them to fold and modify proteins more similarly to human cells. This can produce insulin that is easier to extract, purify, and more similar in a structure to natural human insulin.